So let's talk about the second stage of photosynthesis, which is known as the light-independent reaction. This second stage of photosynthesis has been given many names. Sometimes it used to be referred to as the dark reaction, which is a little bit much because it just doesn't require light. It's not necessarily that it occurs in the dark. Now it's often known as the Calvin cycle, after a guy named Calvin that discovered much of it. It was originally known as the Calvin-Benson cycle, but at some point poor old Benson just got knocked off the end and it just became the Calvin cycle. But we are going to refer to it as the light-independent reaction. It occurs independently of light, but that doesn't mean that it doesn't require light, because it does, because it requires the products of the light-dependent reaction in order to function. So let's just check that we remember the products of the light-dependent stage. Those products are reduced NADP, which is a coenzyme that carries hydrogens from one place to another. So it's taking the hydrogens from the light-dependent stage occurring on the thylakoid membrane to the stroma, where we'll be doing the light-independent stage. So all of the enzymes involved in the light-independent stage of photosynthesis are dissolved in the stroma of the chloroplast. The other thing that we made during the light-dependent reaction was ATP. So we're going to be using both of those things here. And what are we going to produce? We're going to produce sugar. So think back to that photosynthesis equation. We know that we need water, which we've already used in photolysis in the light-dependent reaction, and carbon dioxide. It's here in the light-independent reaction that we're going to use carbon dioxide. But let's just do a little bit of thinking logically. We want to make some sugar, and we know that the chemical formula for glucose is C6H12O6. So how many carbons are we going to need to get hold of? 6, right? So how many carbon dioxides are we going to need to get hold of? 6. So when we think about the Calvin cycle, the only way we can actually make it make sense numerically is if we think in terms of sixes. You need 6 turns of the cycle to make one glucose molecule, because you need to incorporate 6 inorganic carbon dioxide molecules into this organic molecule of glucose. So that's how we're going to think about it, okay? It's a cycle. We can really compare this to the Krebs cycle. Do you remember in the Krebs cycle how you had this 4-carbon compound oxaloacetate, and throughout the process you essentially reform oxaloacetate and it goes through a series of reactions? That's exactly what's going to happen here. But instead of having that 4-carbon oxaloacetate, you've got a 5-carbon molecule known as RUBP, or ribulose bisphosphate. So we have, and remember we're going to think in sixes because it's the only way that it will work. We've got 6 molecules of the 5-carbon compound RUBP, and these undergo a reaction with 6 carbon dioxide molecules to produce 12 molecules of the 3-carbon compound G3P, which is also known as glycerate 3-phosphate. So each of our 5-carbon RUBP molecules has joined with the carbon dioxide to produce a 6-carbon intermediate, which is immediately split in a lysis reaction into 2 3-carbon compounds that we call glycerate 3-phosphate. So now we've got 12 of these 3-carbon molecules. This reaction is catalyzed by the enzyme Rubisco. Rubisco is involved in the first major step of carbon fixation, so in the process by which atmospheric carbon dioxide is converted into the first biological molecule of this process. Rubisco is probably the most abundant enzyme on Earth. So when we think about all these enzymes that exist, Rubisco is the most common because it's so concentrated inside plant cells. And the reason we've got so much of it is because it's not actually that efficient as an enzyme, so we need lots and lots of it. And not just is it the most common enzyme, but it's actually the most abundant protein in leaves. So Rubisco, this enzyme, accounts for about 50% of leaf protein in most plants, which is pretty crazy. So let's keep going with the cycle. Each of these G3Ps is then phosphorylated using an ATP molecule to donate an inorganic phosphate. This intermediate is then reduced. So reduced NADP from the light-dependent reaction comes along and donates its hydrogen. This produces triose phosphate. Phosphate was phosphorylated and it was reduced. So we used the ATP and the reduced NADP from the light-dependent reaction. These triose phosphates now have two possible options. So we've got 12 of them, each one containing 3 carbons. If we combine 2 of those 3 carbon triose phosphates together, we get a 6-carbon compound, and we can convert that into a hexose sugar. We can convert it into glucose. Brilliant. That was the whole point. So we've made our glucose molecule. So we had 6 carbon dioxides come in, and we were able to strip those 6 carbons off and produce our glucose molecule. But that leaves us with 10 molecules of this triose phosphate left over. Those 10 molecules of triose phosphate then undergo a series of reactions to return them back to ribulose bisphosphate. So if we just think about the numbers, we've got 10 3-carbon compounds. That's 30 carbons in total. We can split those 30 carbons into 6 molecules of 5-carbon compounds, RUBP. And along the way, we need to add in another phosphate. So we have a second phosphorylation reaction. So another lot of ATP need to come along. How many ATP need to come along? Well, we create from our 10 triose phosphates, we create 6 RUMPs, RUMPs, ribulose monophosphates, and we add a phosphate to each of those, and they become RUBP, ribulose bisphosphate. So we need 6 ATPs to come in, donate their phosphates, and 6 ADPs go away. So if we look at this whole cycle, for each 6 turns, we use 12 reduced NADPs. We use 18 ATPs, and we produce 1 glucose molecule. That glucose molecule can then be used to convert into all the other organic molecules that we find in plants. We can convert it into fructose and sucrose and maltose and cellulose and starch, and then we can convert it to other things. We can add some nitrogen and we can make some amino acids and proteins for the plant, or we can convert it into fats and oils and waxes, and every other biological monomer and polymer you can think of can be produced from this glucose molecule that we have produced in photosynthesis. It's pretty cool. So the light independent stage doesn't directly require light, but it does need light, because in the absence of light, there will be no reduced NADP. There will be no ATP. And without those two things, you are unable to move from this second stage, from this G3P stage. So you would just get stuck there. All of your RUBP could be converted to G3P, because you just need carbon dioxide for that stage. But then it just won't go any further because you can't convert that into triose phosphates, and you certainly cannot regenerate your RUBP. So light is necessary. So let's just focus on the key stages of the light-independent reaction, or the Krebs cycle. The first thing that happened was that we fixed our carbon. We fixed our carbon into glycerate 3-phosphate. Then we reduced the molecule to produce triose phosphate. This triose phosphate had two paths. It either split off and produced glucose, which then got involved in lots of other metabolic reactions, or it was regenerated back into RUBP. So we have this fixation, reduction, regeneration stages of the light-independent reaction. So take your time to get your head around that, to have a go at drawing it, and well done.